Mitochondria, singular mitochondria, unless specifically are not easily visible under the microscope, variable depending on the physiological activity on the cell. In terms of shape and size, also considerable degree of variability is observed. Typically, it is U-shaped or cylindrical, having a diameter of 0.27 micrometer, average 0.5 micrometer, and length 1.0. Each mitochondria is a double-stranded membrane with the other membrane and the inner membrane dividing it. Lumen distinctly is filled with the dense homogeneous substance called the matrix. The outer forms the continuous limiting boundaries of the organelle. The inner membranes form a number of enfoldings called cristae. Towards the matrix, the cristae increase the surface area. The two membranes have their own specific enzymes associated with the mitochondrial function. Mitochondria are the site of aerobic respiration. They produce cellular energy in the form of ATP, hence they are called the powerhouse of the cell. The matrix possesses single circular DNA molecule, a few RNA molecules, ribosomes, 70S, and the components required for the synthesis of protein. The mitochondria are divided by fusion. Plastids. Plastids are found in all plant cells and in euglenids. These are easily observed under the microscopes as they bear some specific pigments, thus imparting a specific to the plants based on the type of pigments. Plastids can be classified into chloroplast and eugoplast. The chloroplast contains chlorophyll and carotenoid pigments, which are responsible for trapping light energy essential for photosynthesis in the chromoplast. Fat liver carotenoid pigments like carotene, xanthophylls, and others are present. This gives the part of the plant a yellow, orange, or red color. The leucoplasts are the colorless plastid of varied shape and size with stored nutrients, amino carbohydrate, example potato. Elioplast stores oil, fats, and whereas protein. The elaioplast stores protein. The majority of the chloroplast of the green plants are found in the mesophyll cells of the leaves. These are lens-shaped oval, spherical, discoid, or even ribbon-like organelles having variable length 5 to 10 micrometer and width 2.4 micrometer. The number varies from one cell of the cladoma alga to 20 to 40 per cell in the mesophyll. Like mitochondria, the chloroplasts are also double membrane bound. Of the two, the inner chloroplast membrane is relatively less permeable. The space limited by the inner membrane of the chloroplast is called the stroma. A number of organized flattened membrane sacs, like called the thylakoid, are present in the stroma. Thylakoids are arranged in stacks like the piles of coins called grana, or the integral thylakoids. In addition, there are flat membranous tubules called the stroma lamella connecting the thylakoids of the different grana. The membrane of the thylakoids encloses shapes called a lumen. The stroma of the chloroplast contains enzymes required for the synthesis of carbohydrates and proteins. It also contains all double-stranded circular DNA molecules and ribosome. Chlorophyll pigments are thylakoids. The ribosome of the chloroplast are smaller, 70s, than the cytoplasmic ribosome, 20s. Ribosome. Cytoskeleton, an elaborate network of filamentous proteinous structure consisting of microtubules, microfilaments, and intermediate filaments present in the cytoplasm is collectively referred to as the cytoskeleton. The cytoskeleton cells are involved in many functions such as mechanical support, motility, maintenance of the shape of the cell. Central cell, radial spoke, central microtubule, plasma membrane, peripheral microtubules, doublet, interdoublet bridge. Cilia and flagella. Cilia singular cilium and flagella singular flagellum are hair-like outgrowths of the cell membrane. Cilia are a small structure which works like oars of either of the cells or the surrounding fluid. Flagella are comparatively longer and responsible for cell movement. The prokaryotic bacteria also possess flagella, but these are structurally different from that eukaryotic flagella. The electron microstructure of cilium or the flagellum shows that they are covered with plasma membrane. Their core called the axoneme possesses a number of microtubules running parallel to the long axis. The axoneme, their core called the axoneme. The electron microstructure cilium shows that they are covered with their core called the axoneme possesses a number of microtubules running parallel to long axis. The axoneme usually has doublets. The axoneme usually has 9 doublets of radially arranged peripheral microtubules and a pair of centrally located microtubules. Such an arrangement of axonal microtubules referred to as the 9 plus 2. The central tubules are connected by the bridge, is also enclosed by central sheath, which is connected to the one of the tubules is peripheral doublet by a radical spoke. Thus, there are the 9 radical spokes. The peripheral doublets are also interconnected by linker. Both the cilia and flagella emerge from centriole-like structure called the basal bodies.